Well, thanks first to Twist for organizing this conference. So my name is Michael Wiley, and CEO of Praisins Bio, and I'm going to be talking about what our company has been working on developing a universal pan-pathogen infectious disease diagnostic. So one of the first questions we typically get is usually about our name. So Praisins is Latin and means being present. So the Praisins concept started while one of our co-founders, Rudy Powles, was traveling back from Sierra Leone after assessing how diagnostics were being administered during the 2013 to 2016 Ebola virus disease outbreak. He saw an immediate need for a truly mobile laboratory that could decentralize healthcare by bringing the lab capacity to the community that needed it. So the Praisins Foundation was started and a prototype mobile lab was designed in 2016 and then field tested in Senegal with collaborators from the Institut Pasteur in Dakar in 2017. The success from this pilot project led to the establishment of Praisins Care and the first step towards our larger fleet of mobile laboratories that would work with dedicated health centers as hubs, but would help to decentralize the system by being able to go directly to the patient or the outbreak of concern. So now if you think of Praisins Care as the hardware, Praisins Bio is kind of the software. So we are developing a universal pan-pathogen diagnostic using a hybrid capture sequencing. So I was first introduced to hybrid capture by Gary Schroe from Lumina back in 2015 when I was working at the U.S. Army Medical Research Institute of Infectious Diseases in Frederick, Maryland. Our lab had recently sent personnel and equipment to Liberia to assist in characterizing the Ebola virus disease outbreak that was occurring. We tested Illumina's filovirus panel using the RNA access workflow at USAMRIID, and then a few weeks later, after a successful test, we sent reagents to Liberia to assist in characterizing a case of sexual transmission. And the success from there, basically I was sold on the power of hybrid capture. Later, Steve Gross from Gary's group designed a pan-viral panel of roughly 600,000 probes and introduced us really to a then up-and-coming biotech company called Twist Biosciences. And so this was a big jump from 3,000 probes in that filovirus panel to 6,000 in the pan-viral panel. So as you can guess, since I'm giving this presentation, that over the years we've had some success using this. So my time at the University of Nebraska Medical Center, where I'm currently still an assistant professor, and at USAMRIID and work with the DoD, I've had success using hybrid capture to characterize multiple viral pathogens with different partners in Africa. From large DNA viruses like monkeypox that we could use the pan-viral panel right out of the box to generate whole genomes from blood samples, to highly divergent viruses like Lassa that we added in additional probes to help cover the large amount of diversity that goes beyond what is captured in only using reference sequences. It was our most recent deployment to the National Biomedical Research Institute, or INRB, in the Democratic Republic of Congo that caught the attention of Rudy in September of 2018. He thought our use of the target enrichment strategy on the iSeq 100 platform would be a good fit for the mobile labs that they are working on. And so in October 2019, the Praisins Foundation, myself through the University of Nebraska Medical Center, Twist Biosciences, and Institut Pasteur in Dakar came together to apply for the Galleon Prize Med Startup Award. Our submission, which is the idea of decentralizing healthcare and developing disruptive diagnostics and surveillance tools to reach anyone in the world, won for the best collaboration dedicated to developing our underserved populations. Roughly 6 months later, we incorporated Praisins Bio to the US-based diagnostic developer and implementing partner. We believe that marrying these two concepts of the mobile lab with the highly multiplex diagnostic sequencing-based platform will create what we call a preparedness ecosystem that will help to standardize and reduce the footprint needed for infectious disease surveillance and diagnostic testing. So the current state of diagnostic testing typically starts with a visit to a health center. So at the health clinic or hospital, the patient will receive a clinical evaluation and the attending physician can make a diagnosis based off of that evaluation or order some other tests based off the observed symptoms. These tests can range from classical microbiology, which would be culturing bacteria or viruses, microscopy, serological tests that will detect the antibodies produced by the patient's immune response, or antigen produced by the pathogen that the patient is infected with. And there's the nucleic acid-based detection, that is going to detect the genetic material such as DNA and RNA from the pathogen. Sometimes you need a combination of these approaches for that final diagnosis. So for this talk, I'm going to be focused on nucleic acid-based detection side of things. So PCR-based tests have been the gold standard for nucleic acid-based testing. The tests are inexpensive, produce a fast time to answer, and can be administered pretty close to the patient, which we call point of care. However, with PCR-based tests, there is a limited number of targets per reaction that can be assessed to maintain an adequate sensitivity. PCR is also highly sensitive to target erosion, which occurs while the pathogen mutates. And this has been observed in the current pandemic, where we're seeing some of the early SARS-CoV-2 assays fail as the virus mutates. And also, if the assay is designed against just the reference sequence and the total amount of diversity of the virus is either not considered or not known, can also result in false positives or loss of sensitivity. Again, loss of virus comes to mind, as it's a very diverse virus spread across a relatively small geographic range. CRISPR-based tests are a relatively new concept, and what I think have a very bright future in diagnostics. They're inexpensive, have a fast time to answer, and can be administered in the point-of-care setting. I've not handled any of these tests myself, but the literature does indicate there's some design and analysis issues that can affect sensitivity and specificity of the assay, which is expected to only improve over time, as it's still a very young technology. These assays can be multiplexed to a higher degree than PCR, but there are still some limitations with multiplexing and, again, target erosion. Next, we have sequence-based tests, which to date there are no FDA-cleared infectious disease-based assays, but there are some emergency use authorized assays now for SARS-CoV-2, which I think is very exciting and a step in the right direction. These tests can be highly multiplexed, have the ability to detect novel pathogens, and you also generate genomic data that can be used to characterize the pathogen. However, this method is more expensive, there's a longer time to answer, and requires a sequencer and other auxiliary equipment that is not point of care. However, the ability to detect a large amount of pathogens, including novel ones, and to characterize the pathogen I think counters some of these negatives. In the future, sequencing is only going to get cheaper and faster, but there's clearly room in the current diagnostic space for sequence-based assays to characterize pathogens and help determine what is circulating, so that these cheaper, faster assays like PCR and CRISPR methods can be utilized most effectively. But I do see a future where sequencing becomes the gold standard for nucleic acid detection. So here I have a standard RNA-based sequencing workflow that will take your sample, your extracted nucleic acid, convert that to double-stranded cDNA, and then you add the adapters or indexes that are specific to your sequencing platform that you're using. And so you can skip the cDNA steps when you're working with DNA and go right to making your sequencing libraries. So I think one of the common misconceptions, and something I have to admit was my first perception when I was introduced to next generation sequencing, is that even though you're getting millions of reads, you're only sequencing a small subset of your sample and not everything that you extracted. So from the start, you're working with a limited amount of space to try to achieve your goal of sequencing the pathogen that may be present. So in order to get enough data from clinical samples, enrichment methods are essential to increase the sensitivity of detection of the pathogens. These come in two flavors. You have physical enrichment. These include filtering, ultracentrifugation, nuclease treatments. These are all done prior to the extraction of the nucleic acid. Example today would be, how a lot of labs are processing wastewater samples for SARS-CoV-2 using some kind of filtering or ultracentrifugation before extraction and detection. The second form of enrichment is molecular enrichment. This includes anything done after the nucleic acid has been extracted. So I'll focus on three forms, which are depletion, targeted amplification, and targeted hybrid capture. So depletion involves removing the host transcripts, and this is mostly host ribosomal RNA. But there's some other overabundant nucleic acid molecules that you can remove as well. This could be done using DNA probes that bind to the RNA and then are degraded using RNase-H, or with probes that are biotinylated and captured unwanted transcripts and leaving behind what you want to sequence downstream. The ultimate goal of these depletion methods is reduce the unwanted sequences in your sequencing space so that you have a better chance of sequencing a pathogen of interest. These are typically accompanied with metagenomic workflows, which are considered unbiased workflow, even with the addition of these depletions, because you're essentially getting rid of known sequences that you just don't want to be part of that sequencing space. You can also target a pathogen using amplification techniques like PCR. A commonly used method right now for SARS-CoV-2 sequencing is based off a multiplex primer design using the web-based program called Primer Scheme. It's also known as the Arctic protocol from the Arctic network that has put together a lot of useful tools for amplicon sequencing on the MinION. The targets of interest, in this case a viral genome, has primers designed across the genome in a user-determined format for SARS-CoV-2 amplicon sequencing. Most of the application are using 400 to 500 amplicon lengths. RNA is extracted, cDNA is made, and then this is separated into two different reactions where you use the pool 1 in one reaction and pool 2 in the other to generate these separate sets of amplicons. Those two amplicon pools are then added together and prepared for sequencing on your sequencer of choice. This is a cheap and fast method for full genome characterization. The last form of enrichment that I'll talk about is hybrid capture. So this is a technique where the sequencing library is enriched using probes so that the sequencing space is dominated by the targets of interest. Lumina's method that I'll be talking about or I've used has gone through a couple of changes since I first started using it in 2015. The original TrueSeq RNA access kit used ligation-based adapter addition and required 2 rounds of enrichment, making this kind of a tedious process of really a long 2 days or really a 3-day process until you load the sequencer. Lumina still has ligation-based enrichment methods, which I believe is now the TrueSeq RNA exome. The name changes a lot. But the kit that we use now is the Lumina RNA Prep for enrichment, which I believe is a little bit more streamlined. It uses a bead-linked transposase from the Nextera Flex line of products, which tagments the double-stranded DNA, adding the universal Illumina adapters, which are then indexes can be added via PCR. That double-stranded DNA for the tagmentation can come from your second-strand cDNA or from genomic DNA, double-stranded DNA. The next step is what we call is where the magic happens. The double-stranded libraries are denatured and then hybridized to biotinylated probes that the user designs for targets of interest. The probes bind to the target and can be up to 15% mismatch. However, at those higher mismatch rates, the sensitivity does get diminished with these what we call off-target probes, as I'll show in some of the next slides. The probe and the single-stranded DNA library complex is bound to magnetic beads that are coated with streptavidin. So the streptavidin and the biotin bind. You can then pull that out with a magnet because of the streptavidin being bound to the magnetic beads. Libraries are then amplified to make them double-stranded again, normalized, and then loaded on the sequencer. I think Lumina recommends around 1 million reads per library with target capture. We've been able to do this on the iSeq. That gives you around 4 million reads with some modifications to the protocol. And we can do 12 to 16 samples per run. With roughly 100 to 300,000 samples, you can, 1,000 reads can give you your answer for that sample. The development of the rapid reagents on the Illumina mini-seq make this complete workflow from sample to answer using a 1x100 read length capable in 24 hours. So sequencing-based pathogen detection is really this balance between specificity and sensitivity. So unlike other nucleic acid-based assays like PCR and the CRISPR that rely on detection of a signal, the sequencing relies on detection of the sequence. So nonspecific or unbiased methods like metagenomic approaches are an advantage since they can detect any pathogen present. However, they do tend to be less sensitive. as depicted by this lower CT value in the visual I have under sensitivity. Again, this sensitivity issue can be overcome with higher depth of sequencing, but then that comes at a cost in reagents and equipment needed to generate those large amount of reads. Amplicon sequencing is a very sensitive method, and again, as SARS-CoV-2 pandemic has showed us, a very useful tool in generating data, and is the main reason why I think we're over 1.5 million submissions to GISAID today, and which will probably be a lot higher once this talk is actually aired. However, amplicon sequencing is also very specific and starts to lose some of that sensitivity once too many targets are added. Hybrid capture is a nice balance between specificity, sensitivity, allowing for millions of probes to target sequences of interest that have the ability to also bind to off-target sequences or near neighbors at that 15% divergence to the nucleotide level. So the sacrifice in time and cost, with time actually getting a little bit better for hybrid capture, I believe, with the new kits that are being developed. But still, the hybrid capture coming at a premium at a cost in terms of sequencing rates and probes is still, I think, worth it. And I'll demonstrate some of those benefits in some of the first experiments we did as a company. So just to give you a little bit of a feel of the generalized workflow of the analysis, so you have your total reads, adapters are trimmed, low quality bases and duplicates are removed. Reads are then aligned to internal controls and the references that the probes were based off of. And then you have a further duplication, assembly-based duplicate removal to give you finalized depth of coverage. So again, this is something unique with hybrid capture compared to amplicon, where the amplicon, everything is a duplicate. To be able to basically remove duplicates, you have to have a UMI or unique molecular identifier incorporated into your amplicon strategy. One of these first experiments that we did as a company was to, again, set up an on-target and off-target detection experiments for SARS-CoV-2. So we used the original pan-viral version 1 panel that, again, Steve Gross and Lumina designed and Twist manufactured. And so this was all made prior to the SARS-CoV-2 pandemic and only had GenBank references to human viruses included, so that the closest matches to SARS-CoV-2 would actually be SARS-CoV-1 probes. And to make this then an on-target panel, we just spiked in SARS-CoV-2 specific primers into that version 1 pan-viral panel, again, which consisted around 600,000 probes. And then we made serial dilutions of the Twist SARS-CoV-2 control in a human background. We used the ATCC SARS-CoV-2 standard to generate a standard curve and determine the copy number of the dilutions. And so at our two highest concentrations, we get full genome coverage. And then at the 48 copy reactions, we start seeing loss of coverage. And at 4 copies, we go down to 9% and 6% coverage in our 2 replicates. So my previous work with hybrid capture was always based on trying to get a full genome. So we never really looked at the utility of this as a detection tool. We were only using it for characterization. So if you think in terms of PCR, that is only detecting a small fraction of the genome, and sometimes the amplicon length is only like 80 to 120, that 9 to 6% is actually not bad. And it's also interesting to note that when you merge the replicate samples together, coverage increases as the on-target probes don't seem to experience a large amount of bias to a particular genomic content. So next we looked at what we called our detection of near neighbors or off-target enrichment. So for our off-target experiment, we just used that same pan-viral probe set that did not have any SARS-CoV-2 probes in it and repeated the experiment with the same dilutions. As you say, we get reduction in sensitivity with only 17 to 25% coverage for the highest dilutions and 8 to 11% coverage at the 600 copies of reaction. And really no coverage at any of the lower dilutions. Again, looking at this, it's not really that bad given the divergence that you're seeing and that we only had SARS-CoV-1 probes in that pan-viral probe set. Also, you have to take into account that only human viruses were considered for that probe set. So if you incorporate some of those bat coronaviruses, you could probably actually increase the sensitivity and get a little bit higher sensitivity for detecting that novel virus like SARS-CoV-2. So as a company, we have already gone through a couple of iterations of panels. So learning what targets work best. We started with a pan-respiratory panel for our first version. We then expanded to additional bacteria and eukaryotic species. In our current version, we have some additional viruses, bacteria, and eukaryotic species, as well as antimicrobial resistance targets. So while we were doing some of the testing on that version 3 panel, we got contacted by one of our collaborators. So in February 14th, 2021, Ebola virus disease outbreak was declared in Guinea. These was the first reported cases of Ebola virus disease since the 2013-2016 outbreak that resulted in almost over 28,000 cases and 11,000 deaths, mainly in Guinea, Liberia, and Sierra Leone. So 3 samples were sent by the Ministry of Health Guinea to the regional WHO reference lab at the Institut Pasteur in Dakar, Senegal, for sequencing. Institut Pasteur Dakar is one of the labs, again, that we collaborate with, and they reached out to us to see if there's anything we could do, as we had processed samples together before using hybrid capture. So working actually under with my UNMC hat, we sent some reagents through our lab, but this was right around the time when there was this a cold snap in Texas that apparently affected all the shipping everywhere, and so the package sat in Tennessee, where the tracking still today says it's in Tennessee. So obviously never got to Senegal. So obviously the story didn't end there, but I kind of wish it did. It successfully just sent reagents there. IPD gets the reagents, generates the data, and we're just a kind of footnote in that whole process. But it's just not how it happened. So as a small business, it does give us a little bit more flexibility than a university or a government. So our backup to the backup was through Praisins Bio, send out our prototype panel that we've been working on and use it for the first time in a real world experience. I didn't even ask my wife at the time, who was 7 months pregnant, if I could go, but luckily one of the scientists at my company, Carla Prieto, who had worked with me at USAMRIID and at UNMC, had been to Senegal before, was comfortable with the folks there, and was able to get on a plane February 28th and help them produce 2 genomes by March 5th that they're able to share with the Ministry of Health Guinea. And on March 12th, the data from the 3 groups that had been working on actually different sets of samples, but coming with the same conclusions, was posted on with the data on the virological.org. So the first thing we did with the genomes was align those to single representatives. So as you see highlighted in red, the 2 genomes aligned with the 2014 Makona variant, which caused the 2013-2016 West Africa EVD outbreak. We then worked with Andrew Rambal's group. As you can see the difference between the trees between slide 1 and 2 here. His group aligned those 2 genomes with over 1,000 representatives from the West African outbreak. And then again, the 2021 Guinean genome shared 10 mutations that accumulated during the West Africa EVD outbreak, indicating that was a continuation of that outbreak. And again, a conclusion that the 2 other groups working in Guinea were able to come up with using, one group using a targeted amplicon approach with ONT sequencing, and the other hybrid capture using a Lumina workflow on the iSeq 100. And so I'll just kind of end again. Probably everyone's sick of hearing about SARS, but I think I'll end with it because I think this timeline that the Washington Post together kind of the testing in the U.S. is a nice backdrop for how sequencing-based diagnostics could already make an impact today, even with the cost and the time to answer associated with sequencing. So it's not much of a secret that the U.S. had a rough time to start COVID testing. And so again, the Washington Post mapped this from the December 2019, when the first cases were reported in China, to April 2020, when the U.S. was still having widespread testing issues. The genomic sequences for SARS-CoV-2 were publicly released on January 12th, and an assay was developed by the CDC, and an EUA, emergency use authorization, was received on February 4th and then sent to public health labs starting February 5th. Immediately, labs identified some issues with this assay, but it wasn't until February 26th that a workaround was announced. So there's 2 events I'd like to highlight on this timeline where I think sequencing could have an impact. The first one is on January 10th when the WHO issued some kind of some guidelines for handling and testing suspected SARS-CoV-2 cases. So when the samples test negative for all the typical respiratory diseases, they recommended sending the sample off to a reference lab with more capabilities. However, if you have the able to have like more this catch-all assay that we're designing that could be deployed at a more localized level, then we'll get answers faster and not waste time with shipping restrictions and MTAs that can hold up samples getting to regional centers that sometimes are outside of the country of origin. The second events that I want to highlight are the February 29th, when only 4,000 people had been tested by the CDC nationwide, and March 11th, when the WHO does declare a pandemic. Methods like hybrid capture, once established in a lab, are easy to modify for new pathogens once a sequence target is identified, or, as we've shown, these older panels can still detect near neighbors. So new probe panels can be swapped in or out without making changes in other pieces of the protocol and without the need for optimization. So sequencing methods like this could have been used to test more individuals and provide a stopgap while cheaper, faster diagnostics have the chance to be properly validated and deployed. Also, that additional data, if it's being generated, could probably have helped the WHO to make a sooner choice about declaring a pandemic. So just in closing, so this obviously this conference was titled Writing the Future. So I apologize to anyone I disappointed who was expecting to hear that we already had a FDA cleared universal infectious disease diagnostic. It is in the works. I think a lot of, there's some other companies working on similar things. So I think the future of sequencing-based diagnostics is bright and will be a key tool in fighting future pandemics. So I'd like to thank all the groups that were involved in this work, the Praisins team, specifically Carla Prieto, who was able to travel out to Senegal and hand carry those reagents, the team at the Institut Pasteur for carrying out the sequencing and their partners in Guinea, and Andrew's team for the helpful analysis and coordinating the virological posts with all the groups. And just another side note, the lifesaver that is, if anyone's used BioRender, I highly recommend it for making presentations look good, and they did not pay me to say that. So with that, thank you for your time. Thank you, Twist, and the organizers for setting up this conference and giving me a chance to introduce what my company is doing. And just really excited about what we're going to be doing the next year, and we'd be happy to take any questions.